for inviting me. And I should mention, because my other employer would be very upset, I actually have a dual affiliation at the moment. I recently got appointed as professor at the Technical University of Munich. And they have the rule that affiliation always goes first. So, note, it's first. I'm fascinated by life, and in particular by how shape emerges and adapts to allow for this amazing functionality that we observe in life. And one key building block of living systems, of higher living systems, are fluid-filled networks. And what is amazing about them is that they're adaptive in their morphology. And as we learned just in the last 20 minutes, networks are crucial. Here, we're looking at much more large-scale networks that are filled with a fluid, and so not diffusive, but rather advective dynamics are what's governing them. When I'm asking how can these networks adapt, we'll zoom in to one of these tubes. And I want to create one idea. And the idea is that flows themselves are information. So just by having a tube that is filled with a fluid that is flowing through that tube, the fluid is exerting a shear force on the tube wall. The flow comes with a hydrostatic pressure that may act on the wall, or it can transport signaling molecules. And that is all happening in a setting where the tube walls are alive, so they can respond to this information. So imagine, for example, that we take such a tube, and there's an increase in shear force, and as a response, the tube dilates. By dilating, the flow in that tube changes. And not only there, because if we now zoom back out into the whole network, in a network filled with an incompressible fluid, flows are globally coupled. So by dilating that one tube, flows throughout the network change. So also distant tubes get a change in shear force. And so you can see how a feedback of flow and network morphology can unfold. And this is precisely what I stand to investigate, the feedback between flows and morphology. And when doing so, I've cast 4 topics that need to be scrutinized. That is the question of how are flows controlled within the network? What is the power in adaptability in this one tube dilating, for example? What is the role of the network topology, the loopiness in the network? And what is the impact of network hierarchy, of having bigger and smaller tubes next to each other? Now, I will give you a little bit of a glimpse into these topics, and I chose basically 2 topics to show the role of theory in these settings. One story where observation triggered theory, and one story where theory will trigger, hopefully, experiments. So when investigating the feedback between a flow and morphology, my group is using a model system that you see here in this bright field microscopy data. And that I will use when I will now tell you a story about flow control. So the system that you see here is a slime mold called Physarum polycephalum that grows as a single giant cell. And you can appreciate Physarum being giant here when it's crawling in this petri dish to find the food sources. This single cell is 0.5 centimeter in size. It has multiple nuclei that are enclosed in a single envelope that is migrating around. And the cell happens to have a network shape. If we zoom into these networks, we see that they're filled with the fluid that's flowing forth and back. The flow will stop at one point and then reverse, and vice versa. And this movie is actually sped up in time so that you can appreciate that these tubes are rhythmically contracting. So every 100 seconds, a tube contracts and expands. What's going on is that these tubes align with an actin cytoskeleton. That's your basically the same proteins as your muscles are built of. And these are contracting the tubes, displacing the fluid that's in the tubes, and thereby generating these flows throughout the networks. Now, when I'm picturing this image of tubes contracting everywhere all the time, the first question that arises is, how are these contractions coordinated across the organism? And so to scrutinize that, we'll turn to a different kind of data where we now look at the organism on a much shorter time scale of 2.5 hours. On these time scales, networks are static, and I can go in and I can measure contractions throughout the networks, and I can quantify the phase relationship of these contractions, and that will then tell me of how they are coupled in time. And so when I now map out the phases, this is what I see. What we see here is a traveling wave of contractions going through the organism. The color code is the phase going from 0 to 2 pi. And you see that this traveling wave matches organism size and is sweeping this organism. This is very reproducible. You always find this traveling wave, this peristaltic wave, to match organism size independent of your data set or your organism size. So this observation now triggered the connection to theoretical models because we identified that this is a peristaltic wave. And we know actually peristalsis quite well. This is, for example, what our esophagus does. And so now we look at it, it's much easier to think about it as in a single tube level. And so what we have to now account for is that matter that is placed at one point, because we have a closed system, somewhere else a tube has to expand to take up that fluid. And so I can account for that by mapping out the phases to go from 0 to 2 pi along a tube over time. And so having that in mind, I can calculate the flows, and I find blue is flow velocity to the left, red is to the right, and I get the shuttle flow that we also observed in the data. Now, when we look at, now this flow pattern seems to be very bad for transport because everything goes back to its initial point because it's a shuttle flow. Turns out if you calculate the transport, it's not so bad, because it's not relying on the advection per se, but on the diffusion. And so that's the key formula that goes with it that I'd like you to learn today. That is Taylor dispersion. So what this formula describes is the transport of a cross-sectional concentration along long, slender tubes. And so this is the advection velocity that basically gets you nowhere. But here you see that the diffusion term has the molecular diffusivity, but it's augmented by a second term, which arises due to particles hopping along streamlines along your tube with a parasol profile. And that's actually what speeds up the transport. And so now having this picture in mind, I can go back to my data and ask, is this really what the system cares about? So I can, for example, I can take the network architecture and I can generalize peristalsis to a network, and I can follow the spread of molecules in such a flow pattern, and I find that it's very efficient. And indeed, the organism really cares about transport by peristalsis, as we can observe by looking at the organism, like letting it to starve for several hours, for 2 to 3 hours. And we see that the organism reorganizes its morphology. And by doing so, if we map out this diffusivity by this flow-driven transport, we observe that the transport capabilities really increase dramatically, up to 40%. So transport really is something that this organism, these networks really care about. And in itself, it's a very powerful idea that really propagated into lots of different directions. And I'm just briefly going to highlight this and show you that the transport with the fluid flow is what is actually being used to transport information across an organism, across the network. It's much slower than what we know from action potentials, but it's basically what's pretty good enough for the system. Transport of molecules is also what's underlying to generate these traveling waves in the tubes that we observe. And it's actually coupled to the morphology and the topology of the network is even sufficient to explain how these contractions coordinate across these really large length scales that we observe of up to centimeters in size. So with that, I hope I showed you how flows can coordinate in a peristaltic ways, scale with network size, and that the key ingredient there is that this long-range coordination is great for transport, but it's even using transport to generate these large-scale coordination. Now, this was an example of how observation triggered an idea of looking into peristalsis, into transport, as the key property of flow networks. Now, I want to show you a story that is very much perpendicular. In a sense, it's very much rooted in these concepts. But now, with regards to asking a question of adaptability, I'm turning to a very different system. Namely, I'm turning to your microvasculature. Not exactly your microvasculature. It's actually, this is a pristine data set from David Kleinfeld's lab. This is the rat cortex, so it's the rat brain. And I'm looking at this highly coupled network and asking a question of adaptability. I'm asking myself how much control relies in a single vessel dilating. And the idea, or the context where this idea is rooted is the following. Your vessels in the brain are lined with muscles. Now, when your neurons are heavily active in an area and they need a lot of resources, they signal to these muscles and they ask these muscles to relax and thereby dilate the vessel. And the idea is that this dilation of this vessel generates higher absorption of resources that are transported with the bloodstream into that area that needs it. So now, I mean, if you look at this vasculature, it looks like hell. Like, it looks very, very complicated. I mean, how come that this is actually a robust mechanism of how you could generate like a higher uptake of resources? So this is the question that we want to ask. And so when doing so, we turn back to this equation of transport. And now here, I'm actually pulling it down to the real equation in 3D. So you see the term of advective transport. In here, you have the radial transport component and the longitudinal transport component. I'm spreading it out, not having taken the cross-section average yet, because I'm going to add a complicated boundary condition, namely that now my particles that are transported with the fluid flow also absorb passively at the tube walls. But again, very much in spirit of Taylor dispersion, I can actually solve these equations by now taking the cross-section average and then deducing an analytical form for the absorption across a single tube of these molecules through this process. Now, this is a closed form, but you see that I'm not showing it to you. The reason why, it's very complicated, big form. But at least it's an analytical expression. And what I learned from this absorption along a tube wall is that it depends on 4 dimensionless parameters. One is the amount of particles that flows in is the one that can be absorbed. This is what's known as the Packley number, the ratio of advective versus diffusive transport. This is telling you something about the absorption with this advection timescale, and this is the absorption rate. Now, if we keep these 2 parameters fixed, we can actually map out the phase space of these dynamics. And this is how it looks like. So now we have the Packley number over here, that's advection timescale over diffusion timescale, and S, this other parameter, is absorption timescale versus advection timescale. And so this is the phase space where any tube can live in as a function of these parameters. And it turns out that the phase space divides into 3 regimes. And in the limiting cases, we can even give analytical expression for the dynamics in these limiting regimes. So, for example, there's this big regime that we call all absorbing, which basically means that whatever flows into that tube basically gets absorbed, because nothing, like the flow velocity is so small and nothing gets flushed through. And then there are the regime that is dominated by advective or by diffusive transport, and we find very different scaling relationships of the absorption as a function of the tube parameters. Now, this is the phase space that is in principle possible. Now, when we now return to this vascular microaccept, the question is, okay, so where does our brain live, right? And so if we map out where the tube vessels, the vessels in the rat vasculature at least, they turn out to be really up to 90% lie in the advective regime. So this sparked now the question, what is particular about the advective regime that, you know, might might tell us why this, our brain cares about it. And so having taken the perspective that vessels dilate if they need resources, we asked ourselves, how much increase in absorption do I actually get when I dilate a vessel? And so we did the following numerical experiment. We took the data set, we calculated how much stuff is going to be absorbed on each individual vessel. Now, we draw randomly a vessel that is either in the diffusive, in the advective, or in the all-absorbing regime. And we dilate it by 10%, and we recalculate, and we look how much does the absorption in that tube increase. And this is the probability distribution that we observe. So what this means is I increase by 10%, I dilate by 10%, I get a 10% increase in supply. This is this line here. So you see that the advective regime really stands out by being highly peaked, highly robust. So despite me talking about all the flows are globally coupled and this is hell of a complicated problem, then all of a sudden in the advective regime, I dilate and I get pretty much out of what I wanted. But this is not a given. Like if you compare, for example, for the all-absorbing regime, this is kind of random. You dilate by 10%, but what you get, you never know. And also to a lesser extent of the diffusive regime. So now the beauty here is we can actually understand these dynamics because we have our analytical formulas. So if we turn back to the absorption in this advective regime, this is the formula that I've given to you. And now I'm plugging in that the amount of particles that will flow into a tube in the advective regime is also governed by advective transport, so basically by the flow rate. So if we plug this into this is very simple arithmetic, and I find that the absorption in the advective regime linearly scales with the radius. So if I increase the radius by 10%, I get 10% increase in supply, as long as the concentration up front of the tube also stays constant. And so if you have a whole network that is dominantly in the advective regime, this is actually true. And so by making sure that your vasculature is really dominated by this advective regime, you make sure that you're in a rust setting where if you dilate by 10%, you could really get a 10% increase of resources where you need it, where your neurons need it. So this is my example of showing you how theory can not only help to explain data, but how it can also inspire. And so this was a story of how much control relies in a single vessel in increasing transport, flux, or absorption. This sort of is inspiring now to think about how can these capabilities also self-organize network morphology. Now, if we take into account that actually, if you look downstream, the dilating vessel, actually all of a sudden the topology of the network does play a role again. And so there are cooperative effects in the network downstream. And how can they actually feed back on the morphology and drive it to self-organize? I'm very much interested in the question of topology, of how, for example, flows can stabilize flow patterns, of how loops can increase the overall flow. I'm very, very much excited about the question of network hierarchy. If you provide a change of flow in a network, it gets embedded in the network morphology, in tubes dilating and other tubes shrinking, which stays constant over time. So the network morphology actually serves as a memory of previous events, a memory that can also really change the function and drive, especially what Fazaram is known for, the organism to solve complex questions. I would also like to use the opportunity here to make a pitch of why we should care about the physics of flow networks. And I'm being very selfish here because I just look at us humans. And I pretty much only want to outline you of how often we have replicated the concept of having tubular networks. And I'm not even going on the cellular scale here. I'm just staying on the organismic scale. So for sure, there's the vasculature. I told you a story about the vasculature. If you look at our brain, that's a pore network. We have the cerebral flow through tubes there. Our bronchi, our lungs, tubular networks. The kidney has a separate network, the nephron. Our whole intestinal tracts, tubes again. Tumor growth, vascularization is a key step. Lymph system, I could go on. This is just a living system, and it's very inspiring to think about how we can take the physics of living flow networks and map them out in biomimetic systems and thinking about self-organizing porous media, smart materials, or synthetic organ printing. And with that, I would love to thank you as an audience and my group in Göttingen, and highlighting here the names of the people whose work I talked about. Thank you.
Well, we have time for questions.
You mentioned the role of dilation, but and you were talking about tail dispersion, but I maybe missed something, which is there is a dynamical aspect to it. And how does that dynamical aspect essentially play a role in changing the...
What do you, like, you mean that there's a time response?
Yes.
And how, so we only looked at the steady state response because the transport is assumed to be very quick. And so if you only have a 10% dilation, like 10% change, the steady state should find the system should relax to its steady state quickly again.
But in vascular networks, especially if I understood you're talking about David Kleinfeld's work, I think that's not at all at equilibrium. This is a very dynamic, right? Your vasculature.
So you have a couple of dynamics on top of each other. Here we are looking at the changes of a vasculature, of a vessel over time. That is really changing sort of the rest dynamics. On top of it, you have effects such as vasomotion, that is like wriggling of the tube wall. You have to think of your brain. It's actually a closed system because of your skull. So when your vessels on the top of your brain are pulsating, this pulsation actually propagates throughout your brain because it's enclosed in this very rigid skull. So these are dynamics that happen on top that we're not taking into account, but we're very much really working closely with the data sets and the flows that David Kleinfeld himself calculated on this excerpt, and just looking at modification when you permanent, like on a long time scale, really dilate a vessel.
In your single-celled organism with its peristaltic motion, the time scale for the peristalsis seemed to be rather long. I think it was hours, wasn't it?
No, the contraction is 100 seconds.
Ah, okay.
And the changes in the morphology of the network is on a time scale of hours.
Yeah. So the way the tube realizes it's in the contracting phase, is it just that it contracts until it's used all of its ATP supply? No. We believe that what's driving the contractions across the organism is actually calcium dynamics. So that was the theoretical work that I just very, very, very briefly brushed over, is if you model these tubes as an actin cytoskeleton lined around a cytoplasmic fluid, and now take into account that calcium drives the contraction, but also the expansion of tubes drives inflow of calcium via calcium channels, and then the calcium is actually being transported with the fluid flow. This alone is sufficient to explain how traveling waves can form, but much more strikingly how these traveling waves can scale with the size of the system of up to centimeter scale, which is non-trivial, and that's due to the transport with the fluid flow. Okay.
Are there some other questions? Anybody want to ask about maze solving? Then I will. So can you account for the maze solving and Steiner tree minimization, all these things that Physarum can do, based entirely on the representation that you're doing? So when you take the network and you put down a drop, okay, so the maze. So Physarum is known to solve the maze. So here's the downer. Because if you think about like an organism finding the shortest path through a maze, you think about like, I don't know, me being at one entry and trying to find the other entry. Here's the downer. This is not what happens. So what happens is there's a maze, and the maze is fully filled with Physarum that's all connected. And now I'm marking these two exits by putting down food.
So now my work shows if you take a network and you put down food, the food is sort of generates signaling molecules in the network that are transported with the fluid flow and hijack the fluid flow by increasing the amplitude of contractions wherever they are. So that means that those tubes that are sort of along the shortest path, they get larger flow because they get more of this yummy signaling molecules. And so now the only ingredient that you need to add is the idea that large flows get a tube to grow, whereas lower flows get the ones to shrink. So now you see how it solves the shortest path, because the tubes that get the yummy stuff, they are along the shortest path because they get a mod, and so they grow and the others shrink and the shortest path remains. Yeah.
Great, thank you. Okay, so let's thank Karen again, and also